As mentioned, my name is Guadalupe Huerta-Acuchi, and I worked in the Ferreira lab. Great pleasure, by the way. And this is my presentation, a pathway for liquid biopsy, detecting somatic variants in cell-free DNA from pituitary neuroendocrine tumors. So a little background, my project again was on pituitary adenomas, but to kind of understand, we kind of need some background info. So the function of the pituitary gland is to regulate the secretion of the hormones from the endocrine glands, and the hypothalamus, which can be seen right here, is what connects the brain and the pituitary. And pituitary neuroendocrine tumors, or PITNET, arise from the epithelial cells of the anterior pituitary, which can be seen right here. And sometimes functioning pituitary tumors give rise to the oversecretion of hormones such as prolactin, growth hormone, and ACTH. And the primary treatment option that we have for pituitary adenomas is surgical resection, followed by drug therapy. Despite its benefits, chemotherapy only affects the hormone secretion and doesn't do anything in terms for tumor recurrence and doesn't resolve the tumor itself. So here we have cell-free DNA and liquid biopsy. Cell-free DNA is defined as fragmented DNA that is released into the bloodstream by cells that have undergone forms of cell death, such as necrosis and apoptosis. Liquid biopsy, as defined by our lab, since we use blood, is a procedure designed to diagnose if cell-free DNA is being released into the circulatory system by the tumor. Our overall project goal was to see if we can detect tumor-derived somatic variants in cell-free DNA, and this is significant to the field of medicine because it will allow for cell-free DNA to serve as a biomarker to track prognosis and predict therapy response in a non-invasive manner. Considering that surgery is just not always the best resolution for those who have pre-existing health conditions that might face complications during surgery. And detecting tumor-derived variations in cell-free DNA will help researchers to better understand mutation in the body and direct healthcare professionals toward other therapy options that actually target these biochemical pathways that fuel the tumor. So our methods. First, we extracted the DNA from the tumor, and then we did strict tube plasma prep. Practically, we just collected the patient's blood and we centrifuged it. And this separates the plasma, which is this top layer that is seen right here. And it also separates the buffy coat and the erythrocytes. The buffy coat is made of leukocytes and platelets, and erythrocytes are just red blood cells. The plasma is then removed and frozen until later use, and that leads us to our cell-free DNA extraction. So we extract the cell-free DNA from the plasma by using heat and chemical buffers that call the lysis and digestion of proteins and other biomolecular materials as well. And we quantified the amount of cell-free DNA in nanograms per microliter. So here we have next generation sequencing of the whole exome. This consists of 4 primary steps, such as sample prep, cluster generation, and data sequencing and data analysis. And the process can kind of be seen here visually. So here are our results. Our cohort, even though I specifically just did my study on pituitary adenomas, we did have a whole cohort that we sent in. So our cohort consists of 14 tumor samples, of which 8 were pituitary tumors, 4 were meningiomas, and 2 were craniopharyngiomas. And these can all be seen right here. 3 samples are corticotrophs. 1 is non-functioning, therefore it's not actively secreting ACTH. 2 samples are functioning somatotrophs that secrete growth hormone. Sample 4725, which can be seen right here, is a non-functioning gonadotroph, and therefore it doesn't secrete follicle-stimulating hormone. And sample 4891, which is right next to it, does happen to be functioning and also secretes luteinizing hormone. As can be seen in this whole graph, we found numerous common single nucleotide variants in both the tumor and the cell-free DNA. So why are we interested in SNVs? Well, we're interested in SNVs in the exonic regions of a gene because these encode mRNA, and these will eventually be translated into a protein. The second reason why we're interested is because they result in missense mutations. And that means they just cause an amino acid change or non-synonymous mutation. And these may alter the function of the gene, potentially fueling the tumor growth. And one sample in particular from our whole was a corticotrope from a 63-year-old female. In 2013, Dr. Ferreira did a partial resection of a non-functional pituitary macroadenoma. And comprehensive resection was not feasible due to the substantial involvement of the arteries, especially or particularly the left internal carotid artery. In 2019, the patient presented with occasional dizziness, headaches, abdominal pain, and diarrhea. MRI revealed growth of remnant tissue into the stalk, the left cavernous sinus, and the anterior inferior sella. In 2022, the patient returned to the clinic reporting dizziness and blurred vision, and after discussing treatment options with her oncologist, decided to get her tumor resected. Now, this sample in particular was very interesting because it was a silent corticotroph, and it was a pituitary tumor that had recurred, which just makes it very aggressive. Okay, I was like, why didn't it change? Okay, so using IGV, we were able to determine 2 exonic and missense mutations and between the tumor and the cell-free DNA. And these were on chromosome 15 and CSPG4 gene. And these mutations were determined as somatic as they occurred in other cells other than germ after conception. Here specifically, we see that this alanine at position 445 was turned into a valine. And then at position 441, glutamic acid was turned into lysine. Now, this gene is of particular importance because of the pathways it's involved in, such as PDGFR signaling and the MAP cascade, which are really promising pathways and determined to be metastatic cancer pathways. So, yeah. And also, these missense mutations are reported a lot in a lot of central nervous system tissues, especially gliomas and high-grade astrocytomas. So what is next for our project? We hope to continue finding clinically actionable variants since they contribute to the progression of pituitary neuroendocrine tumors. We hope to keep using bioinformatics to aim to find drugs that target these pathways that affect the normal function of these genes, addressing the disparity in tumor target therapy. And we hope to keep conducting experimental validation, including the gene that we had found in the sample, CSPG4, and its potential role in the growth of the silent corticotroph. And acknowledgements, just want to say everybody in this photo, but there's also some missing people. I want to say thank you to all the donors, Dr. Ellenbogen, Mrs. Ellenbogen, all the administrators, and yeah, I just want to say like a special thanks to my lab for believing in me and for all the individuals in this room for supporting me in one way or another. Thank you so much.